Okay. So in the previous figures, we basically found evidence that there's this anti-correlation between transcript length and expression change as these different vertebrates age. We saw that that's also the case in humans with the data that was made available in the literature. And what this group next wanted to do was basically to ask, how do different aging interventions and different mouse lines that have a phenotype of sort of less aging relative to other mice, how do these interventions actually impact that transcript expression relation that we've found in vertebrates? So we start with one example, fetal growth factor 21, which was introduced in excess to different mice. So here we have two different box plots studying two different tissues, adipose tissue and skeletal muscle. And what this group did, or what Stoeger et al. Did, was basically take RNA-seq data that was made available by Murray et al. And they basically binned it into genes that had a relative fold increase in expression and a relative fold decrease in expression, comparing the mice that were given this fetal growth factor 21 compared to the controls, which were given nothing, or maybe some other control. And what they found was that those genes that had a relative fold increase tended to be longer. So it looked like this intervention does tend to ameliorate or basically cancel out that anti-correlation that we see with older mice. Similarly, they found that rapamycin therapy has a similar, basically, correction of the anti-correlation. And we're seeing that in three different groups, one of which did not have statistical significance. But for pretty much all of them, we're finding consistently that the genes that have an increased expression given the therapy tend to be longer. So it looks like both these therapies tend to counterbalance that anti-correlation of transcript length and expression with age. So in the end, they found that after studying 11 different interventions, 7 of them basically counterbalanced that transcript length expression anti-correlation, and 4 of them did not have a significant impact. And that's not to say that those therapies don't work. It's just goes to show that they didn't have an effect on the relation between transcript length and expression with age. Finally, interestingly, they looked at one other intervention, which was an ectopic expression of 3 particular genes, POU5F2, SOX2, and KLF4, which came out of David Sinclair's group with the Liu et al. Paper 2020. And what they found was, first, just looking at 2 different mice, 12-month, which is about middle-aged mice, compared to 5-month, they actually found a sort of the opposite profile. And in this group, they were studying the retina, the ganglion cells. And so they find that in ganglion cells, older mice actually have sort of longer transcripts tend to be increased in expression. So that is sort of opposite to most of the tissues that we'd studied and that we'd seen in the previous figures of the paper. But still, there were some tissues that were observed previously that did have this similar profile. But interestingly, when they introduced these 3 genes with ectopic expression, they found that the therapy actually did correct for this correlation, but in sort of the opposite way that we had seen with the other interventions. And this all went to basically support this idea that these aging interventions have an impact on that correlation between transcript length and aging, whether it's anti-correlation or correlation.